Fantastic. Good afternoon, EEB at all. Welcome everyone to the school year's inaugural Tuesday lunch seminar. This is our first season where we are fully in person. So, if you can't make it here, and if your friends can't make it here, for speakers that allow us to, we will have a YouTube video posted of the speeches, of the talks afterwards. So, our speaker today surely needs no introduction. Though for those of you who have joined us recently, or if you've been living under a rock for a while someplace, we are in the presence of one of the most influential ecological thinkers of our time. And Nate. Dr. John Vandemeer, a third-grade distinguished university professor of ecology and evolutionary biology, Arthur F. Thurnau professor at the University of Michigan. Like many of us, Dr. Vandemeer attended University of Michigan for his PhD. He took a quick trip west, this is all ancient history, to do a postdoc at University of Chicago before returning to us in 1971 as a permanent member of the faculty, and have been with us ever since.
During his illustrious and prolific career spanning 6 decades, Dr. Vandermeer focused on disentangling complex population models in agroecological systems and biodiversity conservation, and most recently, coffee. A core theme throughout these topics is the consideration for both ecological and sociopolitical forces at play, resulting in a well-rounded and ethics-forward approach to science. I could give you a list of the awards with honors Dr. Vandermeer has been bestowed with, but then we would have no time for this talk. So with that, please help me welcome Dr. John Vandermeer and his talk, Applied Community Ecology at the Case of Puerto Rican Ants in Coffee.
Thank you very much. Am I appropriately lavaliered here? Okay. I need no introduction. I don't need to introduce this. What I'm showing here is a model, obviously. It's a cellular autonomous model, supposedly of 12 species in competition with one another. Now, there's different patterns and different colors there represent different species. Okay, it starts with 12 and eventually winds up fewer because they're in competition with one another. This is a spatially competitive system. And as you can see, I hope you can see, maybe when it starts again, you'll be able to see the sort of spiraling effect that it has there. Let me just let it start once more. I think it'll start soon. But this kind of spiraling effect is one kind of pattern that one gets. This is very common. You can find this kind of stuff on YouTube very easily. See the spirals forming there like that. So you get the spiral pattern like that. On the other hand, if you have different parameters, different competition coefficients, you have a matrix of competition coefficients, you change it and you get this kind of pattern, which is really very different. You see these sort of spirals here, and here you see what we refer to as a mosaic. And these patches, they sort of stay constant, okay? Those are two qualitatively distinct patterns, and you tend to get one or the other of those two patterns. Sometimes they're all mixed up, but you tend to get one or the other. One is indicative of what I'll talk about today, intransitive loops. The other is indicative of basically transitive hierarchies. That's the subject of what I'll talk about today, okay? Now, you have basically 3 possible patterns when you have strong competition in a community. And when I'm talking about competition here, I don't want to consider the cases of the fairly obvious, I think, when you have low competition coefficients, you have an equilibrium point, which is a stable equilibrium point that has occupied the thoughts and writings of many, many community ecologists, including myself, for a long time now. But I want to consider the case where we have strong competition. I work with ants, and I've been working with trees in the past. I worked in the tropical rainforests of Central America for many years. And in these cases, my personal prejudice is that competition coefficients in general are very large. You can't fit 2 ants in the same spot. You can't fit 2 trees in the same spot. And so competition coefficients are fairly large. So I want to look at what happens when you have strong competition. Okay, these are the basic patterns that you get. You get one survivor, obviously. This is when you have for example, kudzu or something like that. You have something comes in and outcompetes absolutely everybody else, so this is relatively uninteresting. The other two patterns are the ones that I just showed you. In this particular case, we began with 8 species. Here there's 12 species. Here there are 8 survivors. Here there are only 3 survivors. This is a very typical pattern that you can find. Okay, now the problem here is if these are, imagine that these two maps right here are maps of the place that we work in Mexico. We work in Mexico in a farm, a coffee farm that's approximately 300 hectares big. We have one plot there that's 45 hectares. So imagine that this is our 45 hectare plot, and we see these two patterns in the 45 hectare plot. Well, that's cool. If we see the patterns like this, we would conclude that the top one there is spiral-like and therefore probably includes a lot of intransitivity, and the bottom one is not, and probably is dominated by transitive competition. Unfortunately, to get those kinds of samples, at least in the kinds of systems that I work with it in, is very difficult, because if this would be, say, 100, let's see, 100, this would be 10 kilometers, 1,000 meters by 1,000 meters, okay? And you can't sample the whole thing. If you want to sample each coffee bush, it would be absolutely impossible. So you take a small section. So if you had, for example, say 100 by 30 meters, which is approximately what this would be, and you look at that pattern, you have 3 or 4 species there, yellow, black, and blue, and then sort of a pattern blue there. Would you be able to say if that pattern came from, that sample came from here or from here? It could have come from right here, I think. Or this one, the same thing. It would be difficult to tell which pattern that came from, whether it was a spiral type pattern or a mosaic type pattern. I'll let you decide for yourself. One comes from one, the other comes from the other. If you look at it real closely, you could tell, obviously, because it's very particular in this particular case. But the problem here is that the scale of dynamics, the scale at which the dynamics is working out, which is to say the scale at which you can see all the spirals or the mosaics, is different from the scale that you're able to sample it. In our case, we feel, or I feel anyway, I think my co-authors might feel the same way, but we feel that the scale of pattern formation is very large compared to the scale at which we're able to actually make the samples, okay? So what we have to do is, if we cannot tell from looking at the actual spatial pattern that's there, whether we're dealing with like the top one or the bottom one, what we have to do is we have to use other kinds of information. And in particular, the kinds of information that I feel is useful to make this distinction is information about the natural history of the species of concern, and also time series, how these spatial patterns change over time. Okay, so I'm going to talk about 3 different species of ants. I'm going to let these ants might now refer to these different patterns here. This will be the yellow ant, the blue ant, and the black ant. And let me get rid of that there. That'll come back later, okay? We're talking about Solenopsis, Solenopsis invicta, but Solenopsis is the fire ant. We're talking about Wasmannia, which is the little fire ant, people call it sometimes, or something I call it the electric ant. And Monomorium, I don't know what we call that, the flower ant or something like that. But those are the 3 species of ants that I'll be mainly talking about, at least in the first part of the talk today. This is a photograph of the area that I'll be talking about. This is in the Gran Bate farm in Puerto Rico. And it's basically, it's a farm, it's a coffee farm, but it's an intercrop with coffee and citrus. So from the point of view of ants, you have 3 sort of important places. You have the citrus trees, you have the coffee bushes, and you have the ground. Now, the way these animal organisms fit in here, Solenopsis is down here. Solenopsis forages on the ground mainly, but it also forages on the coffee bushes, okay? But it nests on the ground. Then you have Monomorium that nests only in the trees. It nests in the coffee trees, but especially the citrus trees, and it forages on arboreally, on both the citrus trees and the coffee trees. And then we have Wasmannia, and Wasmannia basically nests and forages pretty much everywhere, okay? On the ground, in the coffee trees, and in the citrus tree. Usually not very high in the citrus trees, but definitely arboreally. Okay, this is The owner of the farm, former owner of the farm, right now, an institute, a nonprofit that we're associated with, just bought this farm, and we're turning it into a sort of an organic demonstration farm, being bought in a land trust, basically to preserve it against the developers that want to come in and build condominiums there. Anyway, this is the owner, former owner, Bernardo, and when Yvette and I first came to this farm back in 2019, as we were sitting chatting with him, we didn't know anything about the farm really, but we're sitting chatting with him. He told us a story. And the story was, the details are complicated, I won't tell you the details, but the story is that he sprayed his farm a year before, and he got rid of this terrible, terrible ant species, Wasmannia. Everybody hates Wasmannia, okay? It stings, and it's a very bothersome, stinging ants. Nobody wants to have Wasmannia around, everybody hates it. So he sprayed a pesticide, and he said he got rid of all of it. Now, Yvette and I had been walking around this little piece of the farm right here, and we had noticed that there was Wasmannia there. And we mentioned to him, we said, oh, well, we saw it. We saw it just yesterday. And he said, oh, damn, it's probably coming back. And so we went out there and we thought, well, it's coming back. It's clearly coming back. Maybe it would be an interesting thing to do. It's an invasive species. It's a chance for us to study the process of invasion of this invasive species. So we sampled a small area. Again, the farm is much bigger than this. This is a small area. And we did see that the green, which he said wasn't there anymore, the green, which is Wasmannia, is sort of spreading over the whole area. We came back 6 months later, and compared to this, then we found this. And yes, isn't that cool? It is that the Wasmannia is moving there. So we said we have an invasion front here. That's pretty cool. And this over here looks like the invasion front. And we're confronted here with these other two species, the Solenopsis and the Monomorium, right? And so we wanted to see what's going to happen next. We came back 6 months later, and this part here now all Wasmannia. You see, just like we were expecting. It came through the invasion front. But the big surprise, of course, was right in here. Right in here, unexpectedly, what happened is Solenopsis starts popping in right there. So what's going on here? Now, unfortunately, just as we're starting to ask that question, what's going on here, COVID came. And so we wanted to sample on a 6-month basis all the time, but we weren't able to. So a year and a half later, we went back to the same place, and what we found was that Solenopsis here, now the red, seems to be taking over pretty much everything, kind of. And then 6 months later, what we have over here, we have Monomorium kind of taking over from the Solenopsis, whereas here the Solenopsis is now moving way into the Wasmannia area, and then 6 months later, moving way into the Wasmannia area, competing here a lot with Solenopsis apparently. And we notice, of course, let me go back for a moment. We noticed here there was some Wasmannia coming in somehow from the other side, so we extended the sampling period and the sampling area, and look what we found. We found more Wasmannia there. So Wasmannia seems to be coming in from here, even as it's being pushed out by the Solenopsis over here. That was July 2022. This is January 2023. The whole process seems to be continuing with Wasmannia kind of taking over from where the Monomorium was dominant, and the Monomorium now moving over here. And then finally, July 23, just a couple of months ago, this is the pattern that we have. So you can see now we have the Wasmannia coming in from this side. We have the Monomorium kind of spreading through here. We have the Solenopsis taking over from Wasmannia over here. So that time series suggests to me, and I hope you agree, time series to me suggests this kind of intransitivity where the Solenopsis is beating the Wasmannia, red beats green. The Wasmannia is beating the Monomorium, green beats blue. And the Monomorium is beating Solenopsis, blue beats red. So I hope you agree. If you don't, I'm going to talk about it a little bit more just now. I hope you agree that we have this kind of spiraling pattern there, which implies that this is an intransitive loop. Back to the natural history now. It's pretty obvious, just from knowing a little bit about the natural history, that the Solenopsis, if you look at the niche gradient of terrestrial to arboreal activity, foraging activity, Solenopsis has this kind of range, Wasmannia has this kind of range, and Monomorium has this kind of range. So you can see who's going to be competing with whom, And then if you look at the nesting site, over terrestrial versus arboreal, you have a similar thing, except it's a little bit more constrained, and Wasmannia has a bigger range of nesting activity. In addition, here's some just random data. There's a lot of data that we have like this. If you take a time series, this is these are minutes here, from 0 to 60 minutes. Blue is Monomorium, red is Solenopsis. And from over a 1 hour period, you see that even though the Solenopsis came here to start with, Monomorium increased and took over. Monomorium increased and took over. And the lower part of the canopy, this is in a citrus tree, why eventually Monomorium took over, even though Solenopsis seems to be pretty aggressive right at the beginning of the time series. On the other hand, if you look at the trunk, this is a citrus tree, you look at the trunk from about here down to about here, why the opposite thing happens. Okay, Solenopsis actually takes over there. So there's some flexibility here, fairly obviously not at all surprising, at least not to me very surprising. Now, another piece of the natural history, an important piece really, is the way Solenopsis actually nests. This is a nest mound of Solenopsis, and this is a cut-through view of what it looks like actually. It has the mound here, which is where all the brood is, and then it has tunnels that go down to the aquifer. But most importantly, it has these foraging tunnels that go out from it. So if you see a Solenopsis mound, fire ant mound here, you just think there's 10 meters that way, 10 meters that way, 10 meters that way, 10 meters that way. They're dominating the ground, okay? They have tunnels that go all over the place that they construct underneath the ground. It's a pretty impressive engineering operation. Now, to study this, what we did is we took a series of mounds, and we put a series of tuna fish baits out from the mound, 10 meters out from the mound. This is my field assistant here looking at the base. And looking at the base, what we find is that as you're looking for the foraging Solenopsises, you don't really find them. You find them at places removed from the nest. This always happens. They're very rarely do they forage around the nest. There's reasons for that. I won't talk about that yet, but there are reasons for that. So what you find is that you don't find them foraging near the nest, but you have these holes that come out from under the tunnels that they have under the ground. You have these holes come out, we call them foraging holes. They just pop out there and they forage around those holes and eventually pop back into the hole to go in the foraging tunnel to bring it back to their nest. And I'm looking at the clock here. I have to talk faster, okay? So on the ground here, what we did, this is an example of the kind of results we get from the ground. This is a 10-meter sampling area with baits positioned every 5 centimeters. And the green baits are where you have Wasmannia. The red baits are where you have Solenopsis. I just separated the two rows here so you can distinguish between the red and the green, the Wasmannia and the Solenopsis, right? Here's a heat map. The heat map is what you saw already from January 2022. And these are all the ground baits that we put out. This is all the ground sampling that we did. And I want to call your attention, you can look at the whole thing, I'll call your attention to this particular one up here. As you can see, in the coffee bushes, which is what the heat map is about, it's all Wasmannia in through here. And even about 15 meters away from any sighting of Solenopsis on the bushes themselves, what we have is on the ground, coming out of their foraging holes, are the Solenopsis. In other words, even though Wasmannia is dominating all this area, underneath the area, what you have is a dominance of Solenopsis. That would become important for a variety of reasons. So let me go on. This all implies that, and I believe strongly implies that, Solenopsis, Monomorium, and Wasmannia are involved in an intransitive loop. That is, Solenopsis beats Wasmannia, Wasmannia beats Monomorium, Monomorium beats Solenopsis. Okay? Other evidence from the laboratory, we have laboratory fights that we do. Other people have done similar laboratory fights. And observations at baits, we find, I had a movie I was going to show you, but I don't have time. Monomorium actually aggressively encountering Solenopsis, and actually, even though it's much smaller than Solenopsis, actually removing Solenopsis from baits by stinging it, okay? So what we have is here's a rock, scissors, paper kind of thing. I also did discover putting this together that there are a lot of Halloween costumes that kids make about the rock, scissors, paper game. This is just one of them, okay? Okay, so what is the importance of all this? Well, first of all, the two of these ants at least are regarded as pests. The third one is some people regard it as a pest, okay? So they're noxious species. All three are invasive in Puerto Rico. But they also are involved, and we've documented this very, very carefully, they're also involved in pest control. Some of the most important pests in coffee are attacked by two of these species, at least two of these species of ants. So they have this contradictory importance associated with them. Also, they're involved, the Solenopsis at least is involved in creating soil structure. Obviously, with all those tunnels underneath the ground, why it's creating porosity in the soil, is creating structure that's inside of the soil. And with that structure inside of the soil is creating conditions for different kinds of root growth and eventually more carbon sequestration, okay? So all of those things are true, and I could give a talk on each one of these if you'd like. But today, I want to talk about something else. I want to talk about how you can unravel some ecological theory, some fundamental ecological theory, using this particular model ecosystem, okay? That was supposed to take 10 minutes, okay? So your introduction took 10 minutes. That's why I'm so far behind. Okay. So, intransitive structures. Now I'm going to talk about some theoretical explorations about intransitive structures. Some of this comes from a paper that Yvette and I just published this year in PNAS. I'll show you that in a moment. But if you can imagine now, the way we normally teach about competition and ecology is, well, not normally, frequently we teach, we begin with a niche axis of some sort. It can be an arbitrary niche axis and fitness on the niche axis, and we have species arranged like this. So species 3, for example, overlaps with species 4, and species 3 have a bigger niche breadth than species 4, so species 3 will beat species 4. Species 4 beats species 5, et cetera, et cetera, et cetera. This is a normal way. I presume everybody's familiar with that from your basic ecology class, or having taught your basic ecology class. We also can have what we call a transitive hierarchy, where the fitness curves are arranged like this. Species 3 beats species 4, 5, and 6. Species 4 beats 5 and 6. Species 5 beats 6, and you get this kind of a structure, where presumably the way I'm casting this at least, these competition coefficients here, x3 against x4, x4 against 5, x5 against 6, are constant and strong, and these can be variable depending on how strong you want the transitive hierarchy to be. Then, in order to get intransitive competition here, you cannot get intransitive competition on a single niche axis. I presume this is obvious if you just think about it for a moment. But with two different niche axes, it's very easy to get intransitive competition. So species 1 beats species 2 beats species 3 on this niche axis, but on niche axis 2, species 3 beats species 1 beats species 2, so you have this intransitive loop. That one right there. These little lines here are meant to be negative. They're meant to be negative symbols, okay? Okay, now, the way the dynamics of an intransitive loop works is you start with 3 species, so you're in a 3-dimensional space, but if you connect the K value, the carrying capacity of one, with the carrying capacity of the other, of another, carrying capacity of the other, what you create is a triangular shape, right? And that is called the manifold. That's the manifold of a system of differential equations, 3 differential equations, which by that I mean, if you have your triangle here, let's see, I don't have a triangle. I was going to bring a triangle, but if you have your triangle here, that means wherever you start, the system very rapidly goes to the triangle. It's up here to the triangle, down here to the triangle. Once it gets to the triangle, then it has a characteristic kind of behavior. In this particular case, you can see the behavior goes from the center of the triangle out and goes out and out and out and gets closer and closer and closer to 1, or then closer and closer to 1, or then closer and closer to 1. That is those equilibrium points, 1, 0, 1, 0, 0, 0, 1, 0, 0, 0, 1. Those 3 equilibrium points are approached, and they're approached in the limit, they're approached, but they're never really gotten to, okay? So this is referred to as a heteroclinic cycle, hetero because they're different points that the cycle is aiming at. So that's a heteroclinic cycle. I'm just going to call it unstable, okay? Because the point in the middle is unstable. There is an equilibrium point, but it's unstable. Contrarily, set the competition coefficients correctly, what you get is a winding, again, you go to the triangle, you go to the manifold, and on the manifold, then you wind in, and you get to a stable equilibrium point there. This is just simply a stable node is the formal terminology of this. I'm just going to call it the stable system, okay? So, again, here's that paper I was telling you about from PNAS. And there's another paper of mine back in the Pleistocene, 2011. It's a long time ago, I know, that is relevant to this also. So what we have is we have this system of Solenopsis, Monomoria, and Wasmannia, as I described. And this is the system of equations that we're going to use. It's an incredibly simplified system of equations. It's a Lotka-Volterra equation where there's only one-way competition. So you only have alpha. You don't have alpha 1, 2, and alpha 2, 1. Here you just have it. Here you have alpha 1, 3, alpha 2, 1, and alpha 3, 2, okay? And your k value, if you like k's, well, your k is equal to 1 and your r is equal to 1 also. So we're trying to study competition, so I set everything else equal to 1 just to study the competition, okay? So what do we get from that? Well, if alpha is greater than 2, why, this is the process that you get a heteroclinic cycle. If alpha is less than 2, why, you get a stable oscillation, okay? And it's very, very nice because qualitatively it pivots on 2.0, okay? So if you're greater than 2, well, you're unstable. If you're less than 2, you're stable. Now, let me remind you, we're talking about competition coefficients equal to 2. In the sort of classic Lotka-Volterra framework, if you have just 2 species, any competition coefficient greater than 1 is extremely large. Okay, so we're talking just about large competition here, heavy competition, right? Only. You can make alpha as small as you want, and this will always be stable. That's true. But even with very, very large competition coefficients, because of the intransitivity, well, you get systems that can be, as a matter of fact, stable. This system, if alpha is greater than 1, very, very strong competition, but less than 2, super strong competition, well, you have a stable system. Okay? Now, the literature is growing on these intransitivities. Some people, like this group from Spain, led by Santiago Solivieres, they're very enthusiastic about intransitive competitions, and they believe that intransitive competition is widespread in plant communities and maintains their species richness, okay? I really like this paper. The reason I like this paper is because it corresponds to my prejudice. If you read the paper, I think you might come to the conclusion that they're exaggerating a little bit, okay? But I don't, because I like the conclusion. Now, contrary to this, you have a whole series of papers, which I think are older, well, it extends back historically more than intransitive competition does, evaluating the evidence for competitive hierarchies in plant communities. There's a bunch of plant competition people who think there are no intransitivities at all, that everything is purely transitive. Okay, so we have a kind of a combination of opinions here, I would say. Now, my feeling is that I think it's probably the case that you have both. If you have a real system out of nature that's relatively complicated, I find it hard to believe that there are not going to be any intransitivities, but I also find it hard to believe that they're all going to be intransitivities. So that means that somehow we have to deal with the question of putting intransitivities and transitivities together in some way that makes sense. Okay, and that's what I'm going to try and do now. And I do this by talking about this, quote, transitive tail. Well, it is a tale about a tail, yes, okay. This is my transitive tale. Now, what we have in our system in Puerto Rico is we have, during that time that we were sampling all that spatial stuff that I showed you already, we found all of these ants. And this is sort of 2019, 2022. I don't have the data here for 2023 plotted yet, but basically the same thing. And we got a lot of different species. Okay, here are just a few of them. Phytoli megacephala, the lion ant, or the big-headed ant. And we have Vandemeer bauri, which is the trapdoor ant, or the trapjaw ant I'm sure you've heard of. We have Phytoli subarmata, which one is a very, very lovely ant. You gotta be willing to you got to be willing to accept the idea that you can be very lovely if you're an ant, okay? It's got a beautiful head, it really does. Okay. Anyway, so I'm going to suggest that we can study this kind of structure. And this kind of structure is, if you recall my introduction into intransitivity versus transitivity, what we have here is we have this intransitive loop, but then Wasmannia competes against species 4, whatever that is. 4 against 5, 5 against 6, etc. We have a transitive tail, which is connected to the intransitive loop. Okay, I'm going to suggest that that's the way nature works, to some extent, you know. So if we examine this structure and look at this structure from a theoretical point of view, this is the set of equations that you just extend from these three here, which were the simple ones, and x4 now. X4 kind of connects these two things together. So x4 has its own special equation, which is the same as x1 through 3. But then you have as many as you want, as many species in the transitive tail as you like. This is anywhere you have for I is equal to 5, 6, 7, all the way to s sub t, whatever s sub t is. And theoretically, at least, I'll tell you, theoretically, it can be millions if you like, okay? Theoretically. Okay, just take this set of equations right here, and we'll look at some time series that come out of these equations. This is just one exemplary time series that comes out of it. If we set the whole thing up so that alpha is equal to 2.1, that's greater than 2, so this is an unstable heteroclinic cycle, unstable oscillation. And you can see how it's going from oscillations here, and the oscillations keep getting bigger and bigger and bigger up to this point right here. I'm showing on the top there the triangular shaped manifolds, so you can see the behavior on the manifolds themselves. And this one is, I've added the transitive chain, the transitive tail right here. Well, I'm trying to indicate with these little diagrams here, this is not obviously not analytical, trying to indicate with these diagrams that we have transitive chains sort of connected to the little peak there, okay? Don't take those diagrams all that seriously. But the dynamics on the triangles, on the manifolds are correct. So we have heteroclinic cycle as we go here, and then here I just change the alpha to 2.0 when the alpha is equal to 2.0. What you have are, for those of you who know the word, centers or neutral oscillations, just like you do in the Lockable-Terra predator-prey equations. You have neutral oscillations. And then if you reduce alpha to 1.9, here you get, as expected, you get the stable point. You move from oscillations here, and the oscillations will eventually go to a stable point. Now if we start the whole thing over, now with all 6 species, with alpha equal 2.1, unstable, right? Unstable. What we get is, well, we get something that looks very similar to the original oscillations, but now you have the green, blue, and the dash line in here that are also in that heteroclinic cycle. And you can keep that going, and somebody goes extinct eventually, all right? But if you reduce that then to 1.9, still very, very strong competition, what you do is that you get this very stable system where all 6 species are maintained in perpetuity in the system at a stable equilibrium point. That's pretty cool, those results. I mean, they really are. They're sort of unexpected. Well, they were unexpected by me when I first started this. They now seem pretty obvious, frankly. But so go back to here. Well, I already showed you this with the foraging exits, okay? So what we did a whole bunch of these transects like this, and the kind of data that you get, here's an example from one of the transects that we got. This is from the where the nest mound is. This is all the way to 10 meters out from where the nest mound was. And these are, over time then, color-coded, green for Phytole megacephala, blue for Phytole subarmata. The colors have changed because I'm talking now about the transitive tail, right? And then the Solenopsis, still in red, is up here. Okay, this is where the nest is. And you'll notice the nest is here, and they're foraging right next to the nest, but then there's no Solenopsis foraging at all until we get to this foraging hole here. And then no Solenopsis here until we get to this foraging hole here. And then no Solenopsis after that. And then you look at the other 2 species, and you see they're kind of foraging those areas where the Solenopsis is not, which is really kind of cool. That happens over and over again, all right? Now, I'm just going to compress these things and put them on a little cartoon diagram, okay? So these green, red, and blue figures here are the same as these 3 down here. They're just all compressed so that I can kind of make my point here. This is the Invicta nest. Here's the tunnel. Here's the foraging holes that they have. And here are the areas where other species can get in there, okay? So what we see with that, then, is that in this particular case, as a matter of fact, what was foraging here is the Phytole megacephala, the big-headed ant or the lion ant. And as a matter of fact, this is where its nest is. So all of the foraging workers came out of here, and they were all foraging in this whole area around here, okay, until they got to the Solenopsis hole, and then they couldn't forage because Solenopsis was there protecting the area. And then further on, that's what the megacephala nest was right here. And then up here, we had a different species, the Phytole subarmata, which is foraging in the gap that Solenopsis left between its foraging exits. Okay? In this particular case, for the study at Grand Bate itself that I showed you all those diagrams for, I showed you the time series for, these are the species that we found in addition to the 3 in the intransitive loop. Now, I'll make the statement that these are all the transitive species. But anybody who knows the system are going to say, how do you know that? And the truth is I have no idea, because we don't know what the competitive relationships are amongst these species. But indeed, these are the species that are not apparently involved in any intransitive loop, but exist in the system, and they exist in those areas where Solenopsis is not. It's a little bit more complicated because the other species are involved also, but I didn't want to get into that in this particular talk. Okay, so now let me go on to the final shot here in this theoretical exercise, and that comes from this particular paper. This is really an excellent paper, I think a really important paper actually, by Levine, Basconti, Adler, and Alesina. And what they were effectively doing, although if you read the paper you don't necessarily see it on the surface, but they're effectively doing is they're taking issue with the current very popular theoretical explorations of a lot of theoretical ecologists called modern coexistence theory. Modern coexistence theory is an incredibly complicated thing with lots of cool mathematics, no question about that. But it has some underlying assumptions. And that's what these people were pointing out here about the 2 of the underlying assumptions. And the first one was that intransitive competitive structures can alter the expectations of the modern coexistence theory. Again, they say that the specific effects of intrinsic competition on coexistence highlight the importance of it, blah, blah, blah. And then, secondly, they talk about higher order interactions. Few empirical studies actually evaluate the ecological importance of these interactions because higher order interactions almost certainly operate in nature. So they were talking about 2 pretty obvious things, at least I think they're obvious, that there are at least some intransitive competition in nature, and we all think, most of us ecologists think that these higher order interactions are probably pretty important in most ecosystems. So we have intransitivity and we have higher order interactions. Now what I'd like to do theoretically, well, before I do it theoretically, let me point out a little bit more about the natural history of what we have here. We do have a higher order effect. It's a pretty important higher order effect. Let me show you a video. Now when I show you the video, I'm going to talk about a fly. Okay, that fly, don't look at that fly. That's not the fly I'm talking about, okay? The fly I'm talking about is actually this one right here, okay? Okay, that's a fly too, but this is the important fly. And so watch the video now. Can you see that little fly running around? See what it's doing? Okay, what it's doing, I hope you can see, but it's attacking the ants. Okay, that's the famous fly in the family Phoridae. And the phorid flies, or some groups of phorid flies, are parasitoids of ants. And the thing is, it creates a behavior pattern in the ant. When it's around, the ant runs around like crazy, basically trying to find a hiding place that where it can get away from the phorid. Now, when you have a piece of tuna fish out, like this right here, sometimes that's so delicious, okay, that you really don't run away, even though the phorid is trying to lay eggs on you. You just sort of sit there and eat the tuna fish anyway. But anyway, they get all bent out of shape. Let me, this is just an update on those graphs that I showed you earlier. You'll see why this is related to the forward in just a moment. But this upgrade shows you this, this transect here, which is there, and then two other transects. This one right here and this one right here. This is the January 2022 parcel that I showed you already. That's the one where I had the, we had the heat map where the three colors were in the heat map, right? And then this one is a year later. The same exact spots a year later, okay? This one especially is important, okay? Because this is the one where you had virtually no Solenopsis anywhere around in the trees, but nevertheless you had on the ground, you had these foraging Solenopsis dominating particular sites on the ground, right? So now in 2023, let me show you what happened in 2023. See, all of a sudden, there still was Wasmannia there, but they're really not very abundant at all. The other two are less interesting, and you can take a look at them if you like, but the ground foraging is an important piece of the intransitive structure. But now, what I want to show you, we did also in this whole area, we did then a survey on the nest mounds and how many of them had forids associated with them. And here's that structure right here. Shaded in yellow are the ones that had forids, and the size is how many times we saw forids. And shaded in gray are the ones that had no forids at all. And you can see a pretty nice pattern here with this area where first of all the Monomorium had control, and now where this Wasmannia is taking over from the Monomorium, why that's where the Solenopsis is being visited by these forid flies. Now the thing about the thing about the forid flies is what the forid flies. I got ahead of myself here. Just a moment. I want to put together intransitivity with the higher order effect now. Okay. The higher order effect being a predator that's interfering with the competition. So Solenopsis is competing with everybody else, but the predator is there and causing it to hunker down. It's typical trait-mediated indirect interaction that people here at the University of Michigan have studied for many, many years. Okay? Now, if we combine those two things together, first of all, let's ask what would happen if we have, on the one hand, Solenopsis foraging really, really a lot. We might expect that we're going to get an equilibrium point here. But then if we decline the Solenopsis foraging a little bit like on this manifold, well, we might get a heteroclinic cycle that is moving out, an unstable situation. So what if we put the two of them together? Pretty obviously, it's going to look something like this, I think, okay? So this is just a diagram, okay? This isn't based on actual numbers. It's just a diagram. Okay, now the thing is, this higher order effect with the predator, what that's going to do is that's going to affect the population density of the Solenopsis, right? So Solenopsis is going to be going up and down right here, right? So we're going to get oscillations. If we look at the forid here and the Solenopsis here, we can expect oscillations here. So we're going to get oscillations between stable and unstable in the intransitive loop, right? So what do you expect to happen? Well, what you expect to happen, before I go into that, there's one more thing that has to do with another intransitivity that's involved. And that other intransitivity, these are the equations now that I've used for that, and I'm running out of time, so I can't go through the details of the equations. They're all very, very obvious Lacavulteri-ish kind of things, okay? But what happens is, based on a lot of work that's been done by other people, especially at the University of Texas, well, we know that the forid actually has a negative indirect trait-mediated effect on having a negative effect on the Solenopsis effect on Wasmannia, okay? So that's pretty well documented. And so we add this effect theta to these equations like that. If you like equations and you know Lacavulteri equations, you will know exactly why those are that way, but I'm not going to explain them. So we do that, and so what do we expect? We expect the Solenopsis to go up and down. Well, it's kind of similar to what we have. Excuse me, I have to go back. This always happens when I'm down to the last few minutes. I have to go so fast I lose my thought. What we have here is an intransitive cycle, as I've been talking about the whole time, but then we have a predator-prey cycle that's connected to it. So what we do have is we have two oscillators that are connected with one another. Coupled oscillators is a really big issue in complex systems these days and leads to some pretty predictable results. And the predictable result that I wanted to talk about comes from this coupled oscillator. Okay, this is an oscillator. There's one pendulum here and another pendulum here connected to that pendulum. So you can imagine now that this would be like the phorid and the solenopsis going back and forth in their oscillations, right? Whereas this bottom one would be like the monomorium and the wasmannia going back and forth in their oscillations, right? Okay, this is, by the way, this is a perfectly good model. It's just as good as differential equations, okay? No matter what your math teacher might tell you. So let's watch what happens with this. It's sort of a classic thing. So we have, first of all, the first oscillator, which is the phorid and the solenopsis oscillating. Now he's going to undo the screw so that the second oscillator starts oscillating also. And that's the monomorium wasmannia oscillator, and you can see what happens. Okay, that's a visual representation of what actually the differential equations really truly do, okay? So as you change the parameters of the differential equation, this is just the wasmannia and monomorium looking at just that space. You get to this situation, which is really interesting, by the way, where this red one here is a chaotic attractor. The blue one is another chaotic attractor. That is in the same four-dimensional space. We have two alternative chaotic attractors, right? And then we can move on where we lose one of the chaotic attractors. I'm changing one of the parameters now. And then it ultimately goes to basically a chaotic system. And here what it looks like in three dimensions. These are the three dimensions of the intransitive component, okay? The classical chaotic structure. And I'm going to leave it at that, okay? I was going to say some more stuff, really cool stuff, but I don't have time because I go all that stuff. And this is really incredibly cool. And I'll just come to this last point here, which I was going to explain through that stuff I just went through. This is one trajectory of a chaotic system right here, okay? You start here and you come to almost the same point here, and then you repeat the whole thing, except it's slightly different. That makes it chaotic. This is a not chaotic point, and the idea here is this looks a lot more chaotic, right? But this is not chaotic at all. This starts here, 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 and here with those four dimensions, and comes out at exactly the same point. This is a periodic point, okay? Okay, so that's it. These are my acknowledgments. Three Fires people, the Anishinaabe people, the Ojibwa, Dawa, Potawatomi, whose land we have stolen and we stand on right now. Specifically acknowledging Kondiarong, whose incredible gift to the Europeans is rarely acknowledged. If somebody wants to know anything about that, you can ask me. And the Taino indigenous people of Puerto Rico, who to this day are completely misunderstood. And NSF funding, USDA funding, and all of these co-workers, which are really all wonderful people that helped a lot with all of this, okay? And I'm just going to leave this up here, just in case you want to read that while I'm answering questions. This is not part of the talk, okay? I'm willing to stay here for hours asking questions. Yes, sir.
So with modifying the Lotka-Volterra equations to get like chaotic nonlinear dynamics, has there been any evidence to suggest modifying in the way to get limit cycles?
Oh yeah, you can get limit cycles fairly easily, yeah.
Yes, but like, is there ecological evidence for modeling them with equations that give rise to limit cycles, as opposed to like centers?
What do you mean ecological evidence? You mean real data from nature? Or?
Yeah, it's like, so I guess I see that you spent a lot of work showing that these models apply to this answer.
Yeah.
But I'm just wondering if a limit cycle could emerge ecologically and that also show up in a model. Is there evidence just?
Well, the limit cycle clearly can show up ecologically in a variety of ways. One way is with a functional response, but there's no functional responses in these equations. Now, when I add, excuse me, I should take that back. When I added the predator, yes, there was a functional response that I added when I added the predator. So there was implicit in the last part that I said, there was implicitly already a limit cycle there.
But the dynamics were periodic?
Oh, yeah. Well, if you get two different oscillators.
I guess I think of chaos and limit cycles as being separate things. Maybe I would.
Well, they are separate things, but if you have two oscillators, if you have a limit cycle here and a limit cycle here, you put them together and very easily you get chaos out of the two of them.
I see. Yeah.
So that's what happened there. We can talk about the details if you like, the mathematical details, which are very interesting.
But like all analytical.
Okay. Yes?
It seems that in this system, at the very least, there are like three dominant species where the most extreme is the intransitive competition, and then you have the transitive tail of like these sort of like rarer, like dominant species. Do you think like that would hold in other systems, like outside of ants, like just like in general?
Well, you're asking me what I think, and frankly, I think that it will. I think that intransitive competition is very, very common, and I think the only reason we don't observe it as being common is because we haven't been looking for it. And the other thing is that ecosystem is very complicated, and you might have an intransitive loop that works for a while, but then something gets in its way. Now, I didn't mention this, but I thought maybe Nate would pick up on it, but there's a new ant that's arrived in the system in the past year. And it looks like it's kind of taking over where we are. We're studying it. I didn't include it in this talk because, well, frankly, I'm trying to ignore it. But it has come in. It looks like it might be actually disrupting this whole cycle. This is a what's it? Technomyrma. And so the last year has just been all over the place, and it seems to be doing something. But I didn't talk about that. So yes, I think they're common, and I think they can be disrupted. And I think you can have 2 or 3 of them, and 2 of them are operated at one time, and the third one's operated some other time. Yes, I think that ecosystems are that way. Certainly with ants, almost certainly with bacteria, almost certainly with plants. What's your field? Huh?
Plants and insects.
Plants and certainly with plants and insects, for sure. No question about that.
I guess, do you think it's more likely though, like with dominant species where the competition is intransitive?
That's a good question. I mean, the whole idea here is that if you have intransitive structures with weak competition, you can't have an intransitive structure, but it's totally uninteresting. There has to be strong competition. So if your rare species are not involved in strong competition, well, then I would expect they're not involved in intransitive loops. But I'm not really sure. That's a good question. I really have to think about that. I don't know the answer.
Why was there forage flies in only half the coffee farm, or where you surveyed?
We think there was only one survey. Okay.
Yeah.
We think that that's an accident of history, and we expect that they're going to move to the rest of the farm. That's what we think.
Yeah.
But that's what we think. Yeah.
So my question is about the rock, paper, scissors limit cycle. We saw how the competition coefficients were affecting the speed at which you approached that outer limit. I'm wondering if there are aspects of the model that would control the actual period of going around the cycle, and what the interpretation of those would be.
Well, with the simple model I showed, it's just that the time series goes very rapidly. It goes more and more rapidly as alpha becomes bigger and bigger. Okay, that's pretty clear. But I think that in the spirit of your question, there's little twists that you can add to the model, which add a little bit of realism, where you don't have a heteroclinic cycle, but you have actually a limit cycle that comes directly from the intransitive loop. Okay, so if you have that limit cycle, then how fast you go around that limit cycle is a combination of how strong the competition is, but then how strong the restriction away from the competition is. So it's a potential combination of things. Theoretically, there's all sorts of possibilities. It's a great idea. Okay.
Yeah, thank you.
Anybody else? Oh, I said I'm willing to stay here for hours. Nate.
Let's see if you can stay here for hours. So what if someone came from a background where they thought just about trade-offs in community structure? It seems like they could use all of these data and just frame it around trade-offs that species deal with, whether they're susceptible to forage or not, whether they can forage in trees or not, deal with high temperatures or not. Couldn't all of these data just be driven by trade-offs that these ants are dealing with?
Well, I think, yeah, I think so, but those trade-offs are what lead to intransitivity sometimes also, you know? So you have a trade-off in, you have a species-specific trade-off in where you nest that contributes to this particular structure.
And I guess even your framing of saying there have to be two niche axes for all of this to work is a trade-off in some ways, right?
Well, I didn't think about that, but yeah, that's true. That's absolutely true. There has to be at least two niche axes.
That's right.
Anybody else? Yes.
You may have already answered this, but in which case, I just want a reiteration. But is there any way to predict whether it's going to be a chaotic or non-chaotic cycle? Or is there any like predictor?
Well, from what's it going to be, chaotic or non-chaotic? Actually, not. With the simple model, yeah, because there's a particular parameter there. That goes back to the question he asked. There's a particular parameter associated with the functional response in the predator part of the equation. So if you know that parameter, yeah, you can predict it. But let's be honest, okay? This kind of structure I think this kind of structure probably exists in nature very naturally. And I think there are other parameters that could be involved also. To say nothing of all the stochastic processes that might be involved, intertwined in all of this at the same time, you know? But you know, why don't you study that?
Your comment about intransitivity and bacterial communities is very curious. Like, one of the things
I did that for you, by the way.
I was starting to think about fungi too, and how you could easily put them in pairs in the lab and test very easily whether the transitivity is common. Like, put two species on the same plate and see which ones win out. But then I also think about the complexity of these microbial communities, where, you know, especially in bacteria, like how often they're rarely in this pairwise kind of competitive scenario. But I guess if I have a question, it's like, do you if we're interested in understanding wood decay fungal communities?
Well, it does lead me to a question. How come you haven't done that experiment? One of the classic cases of intransitivity is with microbes, with bacteria. That's Ben Kerr and Marcus Feldman had a really nice paper. I think it was in Nature or Science, one of the two. But there, the interesting part is that in a petri dish, while they have these intransitive cycles going on, but put the same three species in a shaker, a well-mixed system, and the intransitivity goes away. So that brings up another point that was just brought up the other day. Simone brought it up, actually. Somebody brought this up to her at ESA, and that is, you certainly don't need, but how does intransitivity fit in with spatial pattern? And we do have that one experiment, the Kerr-Feldman experiment, to show that if you have no spatial pattern, you can't get intransitivity. Now, we have our system of intransitivity also. We can't put our species in a flask and put it on a shaker, but who knows? It might be the case that outside of the spatial context, it won't be intransitive. But that's, you know, it's something that crying out to be done with fungi for sure.
I was wondering if you could talk a little bit about, you said the ants are pests sometimes, and then
Yes.
they're also pest control, and then all that concept.
Yeah, that's huge. That's a practical point of this, all of this research. I mean, for at least the Wasmannia and the Solenopsis actually eat the coffee berry borer. That's one of the major pests that's there. And it's complicated because, you know, the Wasmannia is really tiny and the Solenopsis is really big, relatively speaking, like that. Well, the berry borer bores into the berry, and it does all its reproduction in there. So that's all its larvae and eggs and pupae are inside of the berry. Then it flies out to try and find a new berry when it's an adult, right? Well, when it's flying as an adult, why Solenopsis catches it and eats it, or takes it back to the nest, you know? So Solenopsis eats it when it's an adult. But Solenopsis can't get into the hole because the hole is too tiny. But Wasmannia does get into the hole, and Wasmannia is an effective predator on the larvae and the pupae and the eggs, okay? So the two of them interacting with one another create conditions where sometimes they're controlled, sometimes they're not controlled. Now, how that applies, and you're probably thinking automatically, what are we supposed to say to the farmer? And what I say to the farmer is very complex, and I don't know what to do, okay? But the other piece of it, and the really important piece of it, is the pest part of it. Because both of these ants, so, excuse me, Solenopsis and Wasmannia, they're both regarded by workers as very, very noxious because they sting. Especially the Wasmannia. Wasmannia, it's pretty evident that Wasmannia, you saw how it forms these patches, right? These really big patches. And when a worker comes in there and starts harvesting the coffee out of a patch, why, they're very tiny. You don't realize they're on your hands or anything, and they crawl all over your body, and they get back into your neck and into your armpits here, and they all sting at the same time, you know? And it feels like a rope burn is what it feels like. It's very, very, very unpleasant. So when the workers start harvesting in those areas, while they start getting stung, and they just walk, they'll go through those areas really fast. They won't efficiently harvest the coffee. So it has an effect, sort of a socio-political indirect effect on production of coffee by they're attacking the workers. So in that sense, they're a really important pest. Now, the good news is that Solenopsis will control them, okay? Potentially will control them, because as you saw from our results here, if you're going to manage the system such that you're moving Solenopsis in there all the time, you can get rid of the Wasmannia. The only problem is if you've ever stood in a Solenopsis nest for a few minutes, what happens if you step on their nest while they crawl up your leg, and they have this irritating habit that they don't sting you right away. They wait till there's a whole bunch of them up to about here, and then they have a pheromone. The pheromone says everybody sting, and so they all sting you at the same time, which is where they get their name, fire ant, because it does feel like your leg is on fire when they do that. Okay, so I say, well, you know, those mounds are pretty obvious. You don't have to step on them. You just have to watch where you're going. So anyway, that can go on and on, by the way, the discussion of that. That's good. Yes?
I've got one more question from the beginning of the talk.
Okay.
When you were talking about differentiating the kind of spiraling regime from the more blobby regime, I'm wondering if there's been any work on quantitative methods to differentiate those, and kind of if so, what flavor, like how the approach that's been used there.
Yeah, there are some really, really cool stuff, as a matter of fact. Using, what is it called? The micro spirals. I can't remember that. There's a pretty, what's that? Wavelet analysis. People have been using wavelet analysis, which is a really powerful technique. With wavelet analysis, that's the only thing I really know of where it's been analyzed. Though there's one, where's your student? There's, one student that was here earlier, she actually was using a fast Fourier transfers to look at the Fourier structure of the oscillations like that. That's another way to do it. I didn't understand exactly what she was doing. Wavelet analysis is really powerful, yeah.
Were those spatial or temporal Fourier transforms?
Spatial.
Okay.
Yeah, spatial, yeah. Okay. Jacob, you don't have any questions?
I'll ask you them later.
Well, thank you all for listening. Okay.